Good afternoon, ladies and gentlemen. My name is Diego Varela. I currently attend John Marshall Fundamental, and I'm a rising 11th grader. And so for my project, some research project, I did the significance of gastrogens as well as the role in inflammatory regulation. I worked with the Michael Elowitz group, and my mentor was Shia. First off, I want to start with an analogy. I was required during my research to read a lot of research reports, a lot of review reports about gastrogens. And I coined this analogy that gastrogen immune response is a double-sided coin. Now, what do I mean by that? When you're usually referring to this, you were talking about positives and negatives. So right here, I listed both positives and negatives. First, starting off with the positives. Gastrogens help target and eliminate pathogens. Pathogens simply are organisms that help spread disease. Think about viruses, fungi, and anything that really spreads diseases. They basically are parasites. They basically are parasites that contracept the host cell. And what gastrogens do is they are part of this inflammatory mediated cell death known as pyroptosis. And they play a very vital role in this. So this is pyroptosis right here. And it usually is activated either through non-canonical or canonical pathways. The main difference is that in non-canonical pyroptosis is activated by LPS, also known as lipopolysaccharide, and canonical pyroptosis is activated through DAMPs or PAMPs, which are damage-associated molecular patterns, as you can see right here. Okay, yeah. So the main difference between the two is that in non-canonical pyroptosis, there is a secondary inflosome activation. It is an extra step for this presentation. There's really no reason why you need to know this. What I really want to focus on here is the activation of caspase 1 by the near the end of the process. So caspase 1 is a protein, basically meaning that it is cutting up a protein during the process known as proteolysis. And the protein that is cutting is gastrin D. So gastrin D is a various type of gastrin. There are various types, just for context. They go A through E. For this situation, we are using gastrin D. So the process of proteolysis, which I referred to before, basically illustrated like this. This is caspase 1 right here. And basically, like cigarettes, it cuts the C terminal of the gastrin and the N terminal of the gastrin apart. And the correct terminology of this cut would be to basically catalyze it, and as well, anyway, let's keep on. This is the N terminal domain. This is basically what is cut off by the end of proteolysis. And this is probably the most vital part of the process. And the reason why I say this is because without proteolysis and cleavage of the N-terminal domain, it directly results in the pore formation of the gastrin as well as the release of cytokinin release. And the reason why this is important is because the gastrin pore formation, it basically sets up all across the cell membrane. And as you can see right here, it slows up due to the fact that there's an intake of water and a release of cytokinin. In this situation, interleukin. So as you can see, there's an interleukin right here, and then it intakes water. And it intakes so much water that it bursts. So this is the end result of pyroptosis. And basically, it is the rupture of the cell membrane, as well as the release of interleukin. So why does any of this matter? Well, the reason why I ask is because this is just a very random process to talk about. However, I do want to make it sure that everybody knows in the audience that without gastrogens, the whole process of pyroptosis will not happen, and the influx of inflosomes and inflammation will not happen. And so that leads us back to the analogy of gastrin immune response. And now let's look at the negatives. So first off, let's flip the coin. Much better now, right? And so now let's look at the negatives. That being first, tissue damage. This is when the immune system mistakenly attacks healthy tissues during pyroptosis, and this is very bad for the body, as well as chronic inflammation. This is the constant and persistent state in where infosomes are produced and is very unneeded and unnecessary. So what are the consequences? I would like to ask some in the audience, what do you think are the consequences of chronic inflammation? I'll give you a hint, it starts with an A. Exactly. So yeah, autoimmune diseases such as rheumatoid arthritis and multiple sclerosis. As you can see in this picture right here, this is inflamed joint line. And this is what happens due to chronic inflammation. This is the reason why rheumatoid arthritis happens when you're older in life, from the ages of 60 and 65. And then multiple sclerosis is the same, where there's a damage of the myelin right here. As you can see, a healthy neuron versus a nerve affected by multiple sclerosis. And so the thing is that chronic inflammation is what is producing all this. So this goes to the goal of my some research projects, and that is to have a better understanding of what gastric inflammatory regulation works by recreating a gastric A3 model. So the software that I used was Primal. Primal is a digital software known as a molecular visualization system. And the reason why I use Primal is because I was able to take a deeper dive into gastric A3 and basically see how it worked. And I use PDB, which is the protein data bank, which is where users can extract certain structures to work in Primal. This is an example of me testing Primal right here. This is a ligand located in a protein called LASR, and these are the polar contacts around it to do stuff. So this is my first figure of gastric A3 bacteria. As you can see, there are many beta strands, that's what the B stands for, and alpha helixes, which is what the A stands for. And this is just a general wide scope of what gastric A3 looks like. Also, I just want to highlight the N terminal right here and the C terminal right here. As I stated earlier, they basically separate during proteolysis, and this is what basically happens. So why did I pick gastric A3? I mentioned earlier as well that there are multiple types of gastromins. So I would also like to ask someone from the audience, why do you think gastric A3 was the unique gastromin that I picked for infosomal regulation? Nobody, okay. The reason why I picked it is because it's a known autoinhibitor. To explain that auto means self, inhibit means stop. It basically stops proteolysis from happening. So we have a figure of proteolysis right here. No, doesn't work anymore. And this is because gastric A3 stays in a dormant state during autoinhibition. Also note that gastric A3 is not cleaved by caspase 1. It is cleaved by other proteases. I just wanted to add that in there. This is the gastric A3 interdomain. This is the primary and secondary areas where autoinhibition happens. And it is between the alpha-1 helix, the beta strand 2, the other beta strand 1, and that is for the primary. And the secondary is between the alpha helix 4 as well as the alpha helix 9, which is now on this right here. So taking a deeper dive into the primary interdomain interface, as you can see highlighted is the N terminal versus the C terminal right here. And I decided to illustrate even further the alpha helix right here, the 2 beta strands, and the other 2 alpha helixes right here to just illustrate where this is in the gastronomy. And so basically what is happening right here is these aminos right here, for context, the letter is a 1-letter abbreviation for an amino. This one right here is tryptophan, and this one right here is phenylalanine. And the numbers are where it is in the sequence. So this right here is tryptophan 49 and phenylalanine 48. And so basically their role in autoinhibition is basically that they go into the specific grooves of the C-terminal domain with combined with a hydrophobic layer. So why is the hydrophobic layer important? Well, because the whole area right here is a hydrophobic core. Like I said before, the hydrophobic core is important due to the fact that the area surrounding it is very hydrophilic. So these aminos around here are basically bundled up and stabilized, staying in this, staying in this state of stabilization, I guess. And the specific aminos right here are aspartic acid 6, aspartic acid 433, arginine 13, alanine 348, and as well alanine 443. And they basically come together and stabilize this auto inhibitor state. This is the secondary interdomain interface. This is the alpha 4 helix compared to the alpha 9 helix. So I'm just gonna right here, right here, right here. And so basically, it is the same thing, except this time it is more of a second stabilization, the same as auto inhibitor state. Right here we have leucine 184, leucine 181, leucine 186, and basically just like in the primary interdomain interface, they are hydrophobic and they connect with the other aminos on the C terminal. So right here you have alanine 412, leucine 416, leucine 409, and isoleucine 359. So that is basically it. This is just a general overview. Basically, gasoline 83 is known for auto inhibition, and this is due to hydrophobic bonds formed by the residues stabilizing the N terminal to the C terminal. And the reason why this is important is because it further signifies the study of inflammatory regulation that is going on. For further, to give further insight into research being done, there is a drug called disulfiram. It is approved by the FDA and it is known to basically regulate and stop interleukin production and basically prevent autoimmune diseases from happening. So that is it. If there's any questions. Thank you for my acknowledgements. Thank you to the Michael Elliott group, including my mentor, as well as thank you to the Caltech Center of Teaching and Learning. Thank you, Mitch. And as well, thank you to all the other residents. That's it.